Welcome back to this next video, and in this video we are going to talk about the non-viral methods or the non-viral vectors that are used for the delivery of gene of interest in gene therapy. The non-viral methods they present certain advantages over the viral methods. The large-scale production and the low host immunogenicity, they are just the two important advantages that the non-viral methods have over the viral methods. Now previously the problem with the non-viral method was the low level of transfection and expression of the gene. However, recent advances in the vector technology that have yielded molecules and techniques with transfection efficiencies similar to those of the viruses. In this particular video, we are going to talk about some of the most commonly used non-viral methods that serves as vector in the gene therapy. One of the important non-viral method is known as the needle method. And in the needle method, the genetic material of interest that is administered through a needle carrying syringe into the tissue or a systematic injection from a vessel. This is actually the representation how this needle method work. If you look at this one, you have got this needle, and this needle it have got the foreign DNA, and through this syringe we are actually going to insert our foreign DNA into the cell of interest. These are actually the holding plates which are holding the host cell, and with the help of this micro injection you are going to introduce your foreign DNA into the cell. In this particular technique, you are going to use a phase contrast microscope, because of course you cannot see the cell with the naked eye. So you are going to use this phase contrast microscope just to use this needle to deliver the foreign DNA into the cell of interest. Now this needle method, it is one of the simplest and the safest method of gene transfer without any carrier into the muscle, the skin, the liver, the cardiac muscles, and the solid tumors. One of the problem with the needle method is that you are using the naked DNA without any carrier. So the nucleases that are present in the serum, they are going to degrade them, and therefore their action is limited to a very minute time. So this is one of the you can say disadvantages of the needle method, that they are using the naked DNA which are degraded by the nucleases in the serum, and they are cleared by the mononuclear phagocyte system. Thereby it needs continuous re-administration of the foreign DNA into the host cell. Another important non-viral method is known as the electroporation. Now, as the term indicate, you are actually making pores. You are going for a poration process, but that poration process is actually through the electricity. You are making the pores with the help of the electricity. So in electroporation, the short pulses of high voltage for a very short time, for milliseconds, they are used to carry a DNA across the cell membrane. If you look at this one, you have got this recombinant DNA. This is your cell where you want to introduce this recombinant DNA, and this is the plasma membrane of the cell. So what you do is that when you apply a high voltage to this particular cell, that high voltage is actually going to make pores in this particular cell, and when pores they are made, actually the recombinant DNA that can enter into the cell, and when they enter into the cell, the pores they reseal, and the gene of interest that remain in the cell, which can be expressed, and it can produce the protein of interest to make the function of that particular cell like a normal cell. The problem with the electroporation is that most of the cell they die following electroporation. Therefore it has a limited use, including its clinical application. You all know that the cell membrane of the cell that have got a particular semi-permeable nature that allow only those particular materials to enter into the cell which are required for the cell. But if you are making pores in the plasma membrane, that is going to disturb that semi-permeability, thereby in the process of the electroporation, and the pores they are made, most of the cell they die out. Another important method that is known as the gene gun or the bioelastic particle delivery system. So the gene gun or the bioelastic particle delivery system, they are referring to one in the same method. In this technique, the DNA that is coated onto the gold particles, as you can see these yellow dots, they are actually the gold particles to which the DNA of interest that have been added or that had been coated. And these particular DNA which is coated on the gold particles, that is loaded into a device known as the gene gun. This is a diagram of the gene gun, how this gene gun looks like. So this DNA which is coated on the gold particle, with the help of this gene gun, that is introduced into the cell of the host or the cell of the interest. So what you do is, if you look at over here, this is actually the interior of a gene gun. Inside the gene gun, you have got this helium gas, and these the desired DNA which is coated on the on these gold particles, they are present inside the gene gun. You put them into this cartridge like over here, and what you do is, when the helium it produces the pressure, with the help of this pressure, these gold particles they are going to get introduced into the host cell. Now most of the time the DNA that is coated on the gold particles, but you can also use the tungsten as a coating material. But the problem with the tungsten is that it can be cytotoxic to the cell as compared to the gold. Gold most of the time is non-toxic as compared to the tungsten. So in most of the gene gun method, the DNA of interest that is coated on the gold, and with the help of this helium pressure, that is going to be introduced into the host cell. Another important physical method that is known as the sonoporation. So you can compare that this with the electroporation. Now in the electroporation, you are making pores in the cell with the help of the electricity. Now in this particular case, you are going to make the pores with the help of the sono. Sono means sound. So you are making pores in the cell with the help of the sound. So in the sonoporation, the ultrasonic frequencies, they are used to deliver DNA into the cell. So say for example, if this is a cell, you are going to make this cell in contact with an equipment which is known as the ultrasonic producer. The probe from the machine that is going to come in contact with the cell, and when you apply that ultrasonic sounds, that is going to make pores in the cell. And when you make these pores in the cell, you can actually insert your desired DNA into the cell, and once that enter into the cell, that can be expressed, and that can actually produce the protein of interest. Now in the sonoporation, normally a frequency of 48 kilohertz that is used to make pores in the cell. So if you compare that with the normal sound frequencies that the human being use, they are from 20 hertz to 20 kilohertz. So if you are using 48 kilohertz, that is going to make pores in the cell, and through these pores you can insert your recombinant DNA inside the cell. Now the method we just discussed, they are the physical methods, the four methods that we discussed: the needle method, the sonoporation, the electroporation, or the gene gun. All of them, they were the physical method. One of the important chemical method that is used for the delivery of gene of interest into the target cell, that is known as the lipofaction. Now in the lipofaction, what you do is that you are using these liposomes. This is the structure of a liposome. Liposome means a body, lipid body, you can say. If you look at the structure of this one, it is made up of a lipid bilayer. These green heads over here, these are the hydrophilic heads, and these yellow lines, they are actually showing you the hydrophobic tails. So you are actually talking about a lipid bilayer. Now what this lipid bilayer do is that in the center of the lipid bilayer, there is you can say a vacuum, and inside this vacuum you can actually insert your DNA of interest. If you talk about these liposomes, the liposome that are used in the process of the lipofaction, they usually carry a positive charge, and as the DNA that is negatively charged, that means that the DNA can easily enter into the liposome. This is just one of the example of the you can say liposome. This is the you can say the plasmid DNA. This one is your liposome. As the liposome, they are positively charged, the DNA that is negatively charged, so the DNA can actually easily fit into the this particular liposome. So when this liposome that is made, which contain your gene of interest or your desired DNA, so is the this particular liposome, its structure is the lipid bilayer, the cell membrane of the host cell that is made up of the lipid bilayer. They can easily interact with each other, and when they interact with each other by the process of the endocytosis, this particular liposome it enter into inside the cell. This is known as the endosome. When this endosome it enters into the cell, that is going to interact with the lysosome. The lysosome is going to dissolve the liposome, and the DNA that will be released. And when that DNA that is released, that is going to enter into the nucleus of the cell. It is going to be transcribed into the messenger RNA, and that will be translated into the protein of interest. So this lipofaction, it is one of the commonly used chemical methods for the delivery of gene of interest into the target cell. So you have got this liposome, positively charged, the plasmid DNA that is negatively charged. They make this particular liposome that interacts with the cell membrane, and endosome is made. The lysosome act on the endosome, releasing the DNA which enter into the nucleus, and it's actually transcribed and translated into the desired protein. So these are just some of the commonly used non-viral methods for the delivery of gene of interest into the host cell. Gene therapy, of course, is a very attractive kind of the strategy for the treatment of the genetic disorders, but there are certain limitations of the gene therapy. So let us discuss some of the limitations of the gene therapy. Now, for some disorders, or you can say for most of the disorders, the gene therapy will only work if you can deliver a normal gene to a large number of the cells. Say, for example, several million of the cell. So if you are introducing that into a very large number of the cell, of course, these genes, when they reach their destination, they are going to be activated, they will be turned on, and they are going to remain on inside the cells. Now the cells have a habit of shutting down genes that are too active or exhibiting other unusual behaviors. So as you are introducing your gene from the outside, you actually cannot control its expression, and if they are going to be overexpressed, the cell is going to shut down those particular genes, and when they are shut down, that means that the protein product of that particular gene that is not going to be made anymore. Therefore the disorder cannot be treated on a permanent basis. This is one of the limitations of the gene therapy. Another limitation of the gene therapy is that if you deliver the gene to a wrong tissue, because these vector technologies, whether they are the viral one or they are the non-viral one, they are not perfect at the moment, and if they deliver the gene into a wrong tissue, they can actually cause health problem for that particular patient. The problem is much serious in the condition if the gene that is delivered into the reproductive cells, like the sperms and the eggs. As the sperm and the eggs, they are the cells which transfer the genetic information from one generation to another generation, and as you are not sure what kind of the consequences this insertion of the gene into the sperm and the egg take. This makes the gene therapy a very risky kind of the therapy for the treatment of the genetic disorders. Another important limitation of the gene therapy is an unwelcome immune response. As we all know that if an intruder such as bacteria or viruses that enter into our body, our immune system gets activated and that particular intruder that is destroyed and removed from the body. Now in the gene therapy, what you're doing is that you are using this vector for the gene delivery of the foreign genetic material, and if they enter into the cell, you can expect an unwelcome immune response, which can be very serious, and in some cases it can even cause death. If I give you an example of this unwelcome immune response, in 1999, one of the patient by the name of J.C. Gelsinger, he participated in a gene therapy trial who had a rare liver disorders. When they introduced the gene of interest utilizing the adenovector into his body, he died of complication from an inflammatory response. So as you are using this adenovector to deliver the foreign material into the body of this particular patient, the vector is foreign, the DNA is foreign, that created a complicated inflammatory response, and that killed the J.C. Gelsinger. After his death, all of the gene therapy trials in the United States for a time they were blocked, they were halted, sparking a much needed discussion on how best to regulate experimental trials and report health problems in volunteer patients. One of the advantages that we discussed in the last videos about the viral vector is that the retroviruses are very good at inserting the gene of interest into the genome of the host. But the problem with the retroviruses was that it was causing this insertional mutagenesis. And this is one of the problem with the gene therapy, one of the limitation of the gene therapy, that retroviruses on one side, they are very good because when they insert the gene of interest into the host cell, and that gene of interest become the permanent part of the genome, which is then transferred to the next generation of the cell. But the problem was the insertional mutagenesis, that the retroviruses can insert their DNA anywhere in the genome, and if they insert that into a gene, an important gene regarding the cell division, that can cause unwanted consequences like the cancer. So the insertional mutagenesis utilizing the retroviruses, that is another limitation of the gene therapy. Now currently the zinc finger nucleases that have been designed, which have the property that they can insert the genome into the safe places in the genome where it won't cause problem. But the zinc finger nucleases, they are still in the experimental trials. If you talk about the gene therapy, it is very expensive, and the pharmaceutical industries or the pharmaceutical companies, what they usually prefer is to make those particular drugs, those particular therapies which can be used by a wide variety of the patient, by a vast number of the patients. The problem with the genetic disorder is that they are extremely rare. For example, we discussed about the Galabra in our last video that was used for the treatment of the lipoprotein lipase deficiency. So if you talk about this lipoprotein lipase deficiency, it affects about 1 person, 1 per million worldwide. So 1 person in 1 million around the world that will be infected by this lipoprotein lipase deficiency. Now gene therapy could be life-saving for these patients, but the high cost of developing a treatment make it an unappealing prospect for the pharmaceutical companies. And I've told you that the price of the Galabra, which was used to treat this lipoprotein lipase, that was about $1.6 million. So that is a very huge amount. Also, developing a new therapy, including taking it through the clinical trials for the government approval, is very expensive. So with a limited number of the patients, to recover from those expenses, developer may never earn money for treating such a rare genetic disorders, and some patient may never be able to afford them. So this is all about the non-viral methods that are used in the gene therapy.